My name is Pascal Charest. I'm originally from Montreal, Canada, and I've been in Tucson working at U of A for a little over 10 years now. Welcome to the Charest Lab. I'm an associate professor of molecular and cellular biology, and I have my lab in the department, and we study the ability of cells to detect chemicals in their environment and migrate towards their source. So this is a process that's called chemotaxis, and we study this process because it's very important in multiple aspects of normal biology, as well as in disease. For example, during the development, cells during organogenesis, they need to find their path, find their way and where to go to develop into the proper organs and the proper places in the embryo. And then also our immune cells, they constantly use this process to detect bacteria and to migrate to sites of infection in the body. And so another example in humans is cancer cells. So cancer cells will detect chemicals in their environment to disseminate from a tumor and migrate in the vasculature and perform metastasis. So in the lab, we're interested in understanding how cells are able to do that, because it turns out that it's a process that's extremely complicated. And we do a lot of biochemistry, of cell biology, imaging, even a collaboration with a physicist to do mathematical modeling of our data. So we really combined multiple approaches to understand how cells are able to do that at the molecular level.
My name is Helena. I'm a third-year PhD student in Dr. Charest Lab, and Dr. Charest is an awesome mentor. She is the main reason that I applied to the university in general. And my project right now is focused on finding G-protein independent signaling proteins involved in chemotaxis. So in our lab, we use this model organism called Dictyostelium discoideum, and we use that to study chemotaxis. And Dicty is this soil-dwelling amoeba, and it goes through 2 stages in its life cycle, and this change from the unicellular to the multicellular is driven by chemotaxis, which is what we study. So in its unicellular form, you kind of can't see it under the microscope, but in its multicellular form, it develops into what's called a fruiting body, and that is what's on the computer screen in front of me, these dark spheres. And inside those dark spheres are spores, and Dicty will sort of develop into this structure when it enters starvation mode. And those spores are stored in the fruiting body until environmental conditions are right for the spores to distribute and start the unicellular cycle all over again.
The lab has multiple broader impacts in terms of outreach and education. We usually take a few high school students in the lab for internships in the summers, and then there's multiple undergraduate students working in the lab also and learning how to do research, and graduate students, of course, conducting their PhD work and then learning how to become independent scientists themselves. And in terms of the science, the research that we do, we're hoping that it will be very useful to inform better therapies to treat cancer metastasis, and as well as maybe also treat inflammatory diseases where cell migration or this process of chemotaxis plays an important role. I hope you've learned a few things and that this piques your interest for the research that we conduct at U of A.